various aspect of traditional medicine, how our Indian community is going to be standardized standardized point of view. And there is a more important aspect of traditional medicine or herbal medicine in any system, and where there is safety and efficacy is the last important parameter. Although the quality control we are doing, the safety and efficacy of the medicine is important. So in my presentation, I am going to cover mainly the efficacy aspect point of view, because the traditional medicine safety aspect has been already clinically it is used, and most of the time it is safe. And but now we want to take our system of medicines to the international acceptance level. Safety and efficacy both are required, but ultimately, if you are going to do some kind of the efficacy study, we are in a position to do the acute toxicity study and subacute toxicity study. Ultimately, the safety perspective also will be covered in that. So now my topic overview is we are going to do some small introductions, and objective and mission of our PCIMH, and life cycle of the herbal medicines little bit I will explain. Then what is the commercial scenario, then importance of why we need to do the pharmacological studies on the herbal medicines, and how this study, evidence-based pharmacological studies, it is playing a role in the quality control of herbal medicines. And finally, what could be the outcome of our pharmacological studies if we are doing on the herbal medicines. Next slide, please. Next slide. So all can be able to understand my English, whether I'm speaking fastly, because most of the time my colleagues will say that I'm speaking very fastly. So if you feel want to slow, I will slow. Can I slow it? So if you are looking at herbal medicine, you know it's used in various treatments, and there are two important our ancient literatures, that is Sushruta and Sharak Samhita. Both are emphasizing the importance of the safety and efficacy of medicine. We need to conduct the studies in the animals before giving to the human to know the concomitant bad effects, what kind of the side effects it is providing. So it is important for any medicine, including herbal medicine. The first priority aspect is safety, and next one is efficacy. And similarly, efficacy is having an equal importance for the better acceptance of the patients and physicians. If the product you are developed or any herbal medicine it is available, if it is a lack of efficacy, that is one of the important lack of efficacy. If it is there, the medicine will not be acceptable by the patient as well as in case of the physicians. Once the patient says that next visit at the time of coming, I'm not feeling good again the same uh, in the disease condition. Our physician feels that this medicine is not working properly, because firstly he will prescribe in the with the view of the things. It will be treated with another 7 days or 10 days. It will get cured, or in case of 1 month of the treatment of the course of treatment. But if he feels there is no improvement in the symptoms, reduction of the symptoms, then ultimately he will also next time onwards he will not prescribe that medicine. There is a possibility, but it depends upon in our herbal medicines, depends upon the patient conditions and disease conditions, and the genomic point of view also it is a playing. That is a very in-depth if you are looking at that also it's a playing a role. Next step is, so our objective of PCIMH, mainly there is a pharmacopoeial publications, formulation publication, standardization. Apart from all these things, one more as an Apex laboratory, one more important aspect is nurture and promote the quality assurance and scientific literatures publications, and mainly also on the drug research on the ASU and H products. So this is our also one of the important uh, role it's playing. But till date, this area is not explored much in PCIMH. But now slowly that area also is now our director is focusing, and we are working on to improve that area also. Next slide, please. Vision, you know that vision. Next, this is the enter, 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 enter. One enter. Give enter, enter. Okay, arrow is not coming. Fine. So if you are looking, it is a life cycle. You can think about the circle. The circle is starting from the production of the raw material, and second one is processing of the raw material. Another one is manufacturing of the medicine. Another one is storage and transportation. And next one, if you are looking at public or customer satisfaction, this is where in case of any drug if you are taking, it will be the important parameters need to be cycle life cycle. It need to be go through it. If customer satisfaction, physician satisfaction, if it is there, safety and efficacy, and there is no adverse reactions in the product. The product is well accepted and it will be used by the consumed by the physicians as patients as well as physicians will start to prescribe more continuously. So in our India, if you are looking production of the raw material, where we have the good agriculture practice on guideline, that guideline is developed by, that is a benchmark by the NMPB and Quality Council of India. There are two organizations where overlooking this aspect. So production of the raw material is ensured in the organic matter without pesticide content and all this. The next one is processing of raw material. Benchmark here is good field collection practice, GFC, good field collection practice, and cleaning of materials by the company. So to avoid stones or any other contaminants, adulterants, any other weeds, everything it will be taken care. That also the guideline it is published by the National Medicinal Plan Board, Quality Council of India. They were monitoring that aspect also. Next one, if you are coming to the manufacturing of medicine, that's a benchmark here. Is that is there is a pharmacopoeia. They have to follow the industry. They have to follow the things. So in the botanical chemistry as well as the microbiological aspect, already you have seen microbiology. Today you are going to see the microbiology parameter also. So that will be, madam, my given time is 10:30. I will complete. So if you are looking at benchmark, it will be taken care of by then good manufacturing practice, the guideline, WHO guideline also there, and as well as they were following it. So here the role of the PCMH is playing an important role. And now this, if we are moving to storage and transportation, also the pharmacopoeia, where self-led studies, stability studies, where maybe some scientists may be taken the lecture on that aspect also. So that is also important. And we have, there is a standard self-life stability studies. There is a guideline has been published to each kind of the class of preparations. For example, uh, they will say yes, there is a semi-solid preparation, tablet, and particular things where we have the gazette notification. The period of self-life also it is established in that particular. And if you're next, if you're coming to all these things, it is coming under the aspect of in case of the manufacturing aspect, raw material to things. Now, if you are making a one straight cross in that, the end is if you are coming to that, you are you are maintaining the quality aspect, certain safety aspect. For example, heavy metal, you are maintaining a pesticide content, where you are maintaining certain safety level, microbiological aspect. If you are looking at streptococcus, all these things, that see, okay, total microbial load. Apart from these safety, these are the quality control aspect safety parameters. Which we can say yes, we cannot able to say one way we can consider that it is playing some, if it is a level is not in a proper manner, safety issues will come. At the same time, if you are looking at these are, we cannot consider them exactly safety point of view. We can consider it in the safety point of view, but real safety where it is coming, if it is going to the human consumption, there is a, each human it is having a different genomic, and each human the molecules which are phytoconstituents which are present in that, or herbal medicine it is present in that, will act in a different way. So which means that there is a variability will be present in the genomic sequence, where you can able to notice the adverse drug effects and safety issues will be the more will be there, and efficacy point of view. Next one is the efficacy. So if you are giving a quality product, but the product is not, you have controlled microbiological parameter, you have controlled all the pesticide content, heavy metal, everything you have controlled. But if you are going to the patient's customer, ultimately where you are not finding any kind of the efficacy, whether even we will consume the product, if any drug product, if there is no effect, you have 2 days fever and you medicine you are taking. So another 3 days also still there is a fever is in 100 degree, it is continuing, there is no effect, whether we will consume, whether we will perceive to consume that medicine again. No. So the quality control, if we are doing to provide a good efficacious product. So at the time of doing a quality control, you have to look about the plant which are choosing in a proper manner, means the phytoconstituents which will be present in a proper manner. So which is very important aspect in this. So where we are going to play a role, where is the lack now here? Safety is there, since it is a medicinal product. Most of the things is the safety is clinically we are using established, but efficacy is still there is a not much explored compared to that of the allopathic medicine, where it is acting due to that polyherbal formulation and multiple phytoconstituent it is present in that. So how we want to know what is the mechanism it is playing a role inside our thing. Earlier we cannot able to do such a kind of the studies. But now we are applying a modern scientific techniques where we can able to study about how this individual phytoconstituents where it is acting like allopathic drugs, we can able to predict and we can able to study them. So that's where our pharmacology section is going to play a role, and we have taken a lead about that certain studies which has been already published, and we are going to implement on that. Next slide, please. Madam, that depends upon the product. If in case of if you are taking mostly single drugs in our traditional system, very rarely we will use. Even if you are taking some surana powders, 2 or 3 will be present in that. So depends upon generally the powder point of view, the manufacturing process is 6 months. The cycle it will get complete, and it will be 2 years will be the for the powder point of view, maximum 2 years the stability point of view. Okay. Up to the things it may be 3 to 6 months manufacturing process. Manufacturing process, if you are looking, maybe 3 to 6 months. If you are taking, if it is well established product, the where the standard protocol already set, the preparation, everything, pilot, everything will be covered, and it will be even 1 month, within 1 month, the productions, everything it will be get over. The cycle already they were maintaining properly. They were continuously they were manufacturing. They were having enough raw material. Within a month, they can able to complete production. For sure, you have all standard the processes are all we have prepared already documented. Yes, they have to good manufacturing practice. They have to maintain all the documents where the raw material preparing, what is the what is the when, which date, how the storage conditions, everything in GMP, good manufacturing practice as per the guideline. They have to maintain properly schedule. So that will be everything. It will be fully controlled. Everything in the guideline is there, fully controlled aspect in this. And for even for the stability study, we have gazette notification. Madam, that notification also is a gazette notification for us. So where we are doing different kind of the formulations, different duration of the stability study period also they given. And stability study, whether Dr. Vijay Gupta has already taken the things now. So he may be displayed that guideline and everything, stability. So this is the general already you maybe came across. Only thing is I would like to now focus on Aruno botanical, organoleptic, physical, chemical, microbiology. Today you are going to see that. I am going to focus only on the biological aspect. That's the safety and efficacy point of view. At present, now we are focusing on the efficacy point of view. And later stage, once our lab it is well established, then we will also will look about the safety studies also we can able to conduct in the advanced level safety studies. Next. So these are the regulatory aspect of our Indian system of medicine as per the Drug and Cosmetic Act 1940. Hope you maybe came across this also. You came across, no? GMP, you maybe came across Section 3. So I don't want to waste the time much in this. But you know that 3A is about the Ayurveda, Siddha, Unani drugs, and 3H is Section 3H is patent and proprietary drugs. Chapter 4A is that is the provisions, license applying, uh, everything will be present in that. Then two main schedule is first schedule authoritative books pertain to the Ayurveda, Siddha, Unani drugs, and second schedule where quality standards for the homeopathy. The homeopathy drugs I am not touching in my presentation. The reason is homeopathy we are using, but it is not an Indian origin medicine. It's German, and it's not an Indian origin thing. So I am not touching that area at present stage, because most of the time in homeopathic medicine also where they were using tinctures, mother tinctures. It is they were using the single drug. Mostly they were preparing from the single drug. It's not like our traditional system of medicine in a combination of 3 or 4 or up to even 10 plants. They were not going to use it. Single plant material, raw material, mother tincture with alcohol. They were going to prepare the things. When based on the mother tincture, they were going for the serial dilution. Depends upon the patient disease conditions. Okay. So that area I'm going to cover, and I'm going to focus only on the authoritative books pertaining to the AACNH and what kind of the thing, whatever the in the authoritative books for schedule. Ayurveda some 58 book is there, books is there. So these are the ancient literatures which have been written by our ancestors, where the particular formulation or medicinal plants is used for the particular disease, to treatment of the particular disease. Okay. So our pharmacopoeia, what they were doing, they were decoding the authoritative schedule books, what are the formulations they were given, and we are developing a standards for them. That is our pharmacopoeia main room. From the base of these authoritative books, they have to take some formulations. That formulations they were going to decode it in the formulary point of view, and in the single drug point of view, they were going to work for the single individual plant. They were going to prepare a standard and what are the tests need to be performed. This is what the things it is main work of the our pharmacopoeia commission. Okay. So now in this authoritative book, there are a number of formulations is present in that, but now we have achieved only small uh, portion of the authoritative book, the formulation has been standardized. Remaining, there is a lot of formulation still it is existing for the standardization point of view also, as well as to study the pharmacological aspect also. Okay, next slide, please. Whether these everything is covered or I want to cover everything, so I can skip this. These are the just I will say that. Uh, that's why I don't want this, but I want to touch that also. Otherwise, the link will not be there. Okay. So these are the rules um, in respect of AI researchers, how they were license they were providing, what kind of studies it is required to obtain the license for the manufacturing of the product as well as the sale of the product. Okay. Next slide, please. So the previous slide. The first one is mainly the authority books. I have underlined authority books of AIC in the first schedule. So which where it is mentioned in the first schedule, as such you are taking a formulation, as such you are doing the procedure, and you are going for the licensing. No need of any testing. Only some published literature is sufficient to get the license in India, in state level as well as state. But in patent and proprietary means you are doing some kind of the changes in the formulation of the authority books formulation. Any single plant if you are changing, or formulation dosage from tablet to capsule if you are changing, or liquid semi-solid preparation if you are changing, you need a studies, certain studies, publications. It is like that. Next slide, please. Next. Okay. These already you are seeing that I said that if 3A directly you are using ingredient as per the text of authority book. Indication is as per the text. For example, if you are having a one formula, the formula as per five plant, the same five plant, same formulation you are using, same indication used in the treatment of inflammation. Suppose you are saying that it is having used in the rheumatoid arthritis, they mentioned in the authorities somewhere. So that's all. You no need of the safety study. Some published literature is sufficient for you. But if you are going to do any kind of the dosage form, sometimes there is a published literature and studies are required. Next slide, please. If any new indication, if you are making, so for that you need to provide a study. Otherwise, as if you are moving with the authority book of the first schedule, as per the Drug and Cosmetic Act, you no need to provide any kind of the study. This is the status in our there is approval process of the thing. So which means that why I am stressing here, the pharmacological studies, safety studies is not required because it is a clinically used in many years. The safety is established, efficacy is established. This is the concept. So that's the reason no need of any kind of the studies where you can directly you will get the approval for the manufacturing and marketing. Next one. The,
What if the regulators are asking for the safety studies? Can you still provide it?
no, actually, previous study see if that I will come to the backside. Means if you are coming back, the slides will be there. What are the studies it is right here? You will come to it. Okay, next slide. Uh, okay, next slide. And one more previous slide you can go. Suppose in our schedule there is a you can look the second one. There is a schedule Ayurveda Siddha Unani drugs with any one of the ingredients of schedule E1. There is a schedule E1 which is mentioned here. There is a poisonous drugs. Okay, for example, cannabis, such a kind of such a kind of cannabis. So these are all where we cannot able to use as such. So that's where the schedule E1. Such a kind of the any ingredient you are going to use as per the text, and if indication as per the thing, and the published literature if it is there, it is a fine. But safety study, some kind of the efficacy study is required for that. If you are not using a schedule given drugs, but you are following as such the first schedule, some formula as such mentioned, as such if you are following, as such you are as per the procedure if you are manufacturing, no need of any kind of the studies, published literature. With that literature, you can provide, you can get the approval. Next slide. These are some kind of what we can say that health promoter kind of the medicines. For that also same. If you are using as such the first schedule book, no need of this. Next slide, please. Now another thing is most of the time when we are using a aqueous extract, hope in depends also where most of the time we will in traditional medicine we will use only the aqueous extract, the plant material boiled in water, the precautions, the precautions we will use, right? So so for that you no need of any kind of the data study because aquaculture is where we are using from the ancient time. So already it is people are consuming. There is no much studies. It is reported safety and efficacy also they were looking. So no need of any kind of the studies. Okay, but nowadays where there is a slightly sometimes if you are looking in our ancient system, there may be fermentation process will go. There is a fermentation process whenever it is goes. Ultimately, there is a production of the alcohol also will be present in there, right? If you're fermenting some medicine, sometimes even they will use some kind of the yeast or some materials. So where they will the fermentation will occur, ultimately alcoholic content will be there, but the content will be very much will be present. So nowadays they are using hydroalcoholic extract also. So hydroalcoholic extract if they are using, where if new indication if you are making as per the formula, but new indication if you are making or you are changing any kind of the ingredient in that, you need to provide a safety and efficacy study, public literature you have to provide to get the approval. Okay. Other than hydroalcoholic extract, if you are using, suppose that we can say it's a fractions, standardized fractions, where chloroform fractions or ethanolic fraction of the thing, such a kind of a standardized extract if you are going to use, you have to provide acute toxicity study, chronic mutagenicity, teratogenicity, and all this and efficacy study also it is effect of the proof of also it is required. Is it clear now? The studies is where it is required. Okay.
How about uh, if it is in a classical drug, it is uh, published in your previous years or in your in the culture. But since we have now a new era of there is already Western uh, medicine. How about do you do study about drug herb interactions that uh, the classical drugs may interact with the one that being taken during this time because those times there is no modern drug that being taken. So yes, that is uh, in those in their times they are safe, but possibly it could be not safe during this time that some people may taking medicines uh, uh, that are just developed within this era.
Okay. I will address you continuation of the slide. It will come. Last I will address your question. So you will come to know what's our aim. Okay. Next slide, please. I will explain you. After the completion of all these slides, where our pharmacology section role, we have future plan everything. Your questions will be get answered there. Okay, I will answer you. So, if you are coming to is a general guideline of the research point of view, all non-clinical studies, clinical studies, raw material you have to collect. We have to follow GMP, good agriculture practice, good field collection practice. You have to make the things non-clinical studies, formulation you have to standardize as per our pharmacopoeia. We have to consider all the parameters. Then you have to come to the clinical studies, and this is a general drug development point of view in herbal medicine also. Next one. Next one. So this is almost the same. What are the clinical safety data is required, not required is the same. If you are using as such a 3A, drug and cosmetic acts section, as such if you are using a classical formulation, you can see that as per the text, as per the text, as per the text. Only last if you are looking, there is a new indication. If you are changing, you need the study is required. Non-clinical efficacy data is required. Otherwise, not required. In pattern and preparative things, As per the text, if you are using, you are giving a same indication. No need of any of the studies is required. Next one. But if you are going to your questions, Mr. your question is where if you are looking at the pattern and proprietary drugs other than aqueous extract and hydroalcoholic extract, if any specified ingredient if you are changing or specific claim if you are making, as such a classical text, no issue, no need to do any study. But specifically, if you are using any claim instead of authorities they mentioned in the textual books, but you are saying that I am going to use for the diabetes or cancer, so where you have to provide the, and you are saying that I have changed one ingredient also in the composition. So now this composition formula where we can able to provide for the cancerous patient disease. If you are claiming that for the going to the licensing authorities, then they will not all this kind of the studies. Oral single acute toxicity study, repeated dose toxicity, reproductive study, genocidity, carcinogenicity, and efficacy clinical data also small kind of the population maybe they may be record. You need to provide. Next page, please. This is the dermal toxicity, photoallergy, all these kind of the studies you are in a position to provide data. Whenever you are changing the formulation, any one, any total formulation you are changing, or any one even single ingredient you are changing, or you are giving a new indication. You are claiming a new indication. So you are in a position to submit the study data. They consider as a new drug. Okay, next one, please. Similarly, patent and proprietary drug also. Next. Okay, now we will, so this up to this where we have seen about the things. Hope you have some clarity, some idea about the things. Now we are coming to the commercial scenario in domestic market. No issue as per our, because each domestic market in Indian country, respective country, where it is playing a respective government cosmetic act and according to the manufacturing process as well as marketing will be sale will be taken place. But if you are going to the international, all our drugs were considered on the food and supplementary category. So where if you want to consider as a drug in Western countries where we need a scientific evidence on the safety and efficacy point of view, it could be either preclinical or clinical. Anyone we are in a position to claim, to substantiate our product is good, how it's working, it's safe. You have to make the things. So monitoring and QC of herbal medicine in the safety aspect, there are nowadays there is a pharmacovigilance program also it is available, or herbal also they are doing. Maybe are just reaction. Next one. So what is our importance of the pharmacological studies next? So you know that pharmacological studies, you will get all the ideas, the route of administration, how much time you have to administer, what is the mechanism point of view, as you asked, no? How the so mechanism, drug interactions. So everything you will come to know. So pharmacological studies are important to establish the safety. So if you are coming to that in case of general aspect, uh, where they will take in allopathic drugs, in silico, some kind of the studies, high throughput screening, then in vitro, in vivo, randomized clinical trials, then it will go 10 to 15 years for the normal conventional methodology. But now, if you are looking in case of our Ayurvedic, our system, traditional system, where already the product is used in the market and the particular kind of the diseases, but the thing is the scientific data is not there. Previously, what happened? The new drug synthesis, to further they were generating a scientific data and they were going for the approval. At the time of approval, they were having all the safety, efficacy, quality control, active pharmaceutical ingredients, what are the impurities will come in the things. All the data will be presented. Okay, so 15 years. So you have all the dossier you have in your hand. But if we, in our traditional system, if you are looking, already human is considering in the clinical two, we are taking into the bench, and now we are going to analyze why this effect is coming. What is the formula in this percent? Which phytoconstituency is responsible for the particular activity? And now we are reworking on the things, and we are saying that yes, this is working. For that, we are using some in silico studies, in vivo studies. Sometimes it required clinical studies. Clear? This approach is called as a reverse pharmacology approach. This is very easy for us because what happened in the conventional methodology, first they will use the disease, okay, and for they have to understand the disease process, and they want to understand why the disease is coming, what is the protein, what are the changes molecular level it is happening, and how to inhibit that. So depends upon that they were developing a molecule, right? So they want to prove that one, they have to do a lot of things. That's entirely time consuming process. But here, already the disease is treated by the thing. So we know, yes, this formula is working for this, but we don't know how this formula is working. So we want to establish that. That's all. Our work is already product is used in here. The taste is there. We want to check why this taste is coming. That's all. But they don't know the taste. They want to prove that yes, because of this thing, that the efficacy is coming. But here efficacy is there. We are need to go back to reverse analysis. We are going to do the things. Okay, next step please. So these are the traditional methodology where new drug development processes. Next step is you maybe know that. See now we are going to approach. There is a two kind of approach we are going to do things. One is system pharmacology approach. Next step please. Next. This system pharmacology approach we are going to use in that. So where is your traditional? Generally, traditional Chinese medicines are they were doing it much better. Their medicine is accepted globally, uh, especially in Western countries. What you ask, Western countries point of view, the reason is they were working in a systematic way, and they were exploring the reverse pharmacology methodology. So how they use traditionally, why it is, how it is working, they were establishing. So they were doing some kind of the phytoconstituents isolation, and they were doing in vitro studies, in silico studies, computer-aided drug design discovery studies. Then they were going to the molecular pathway, which pathway it is acting. Then they were taking into the clinical aspect also. So they were making all this process in a properly. So this is called as a system pharmacology approach. Means the molecule, how it is acting inside of the our body, which one is responsible for that. We are going to study that. That is where your question. It is one part of you. And drug interaction, I will come to the next. Click next. So now this is the one example I have taken the system pharmacology analysis, and our aim also is going to be in our PCIMH. We are going to implement reverse pharmacology approach, and we are going to establish for our Ayurveda, Siddha, Unani drugs, how the molecule is working in based on there is a different methodology, system pharmacology approach. We are going to do the things. Next slide, please. Next slide. So this is where they were doing that common. First, we the same thing the approach we are going to make, but sort of slightly we are going to modify that also. And if you are going to look any herb we are going to take in this formulations, we are going to take it. Compound database first we will develop, then we will screen it. Which compound is potential compound we will analyze using a in silico method. Using a in silico method. You maybe hear the in silico studies. Not yet. Computer simulation studies. We are going to do the things, and based on that, we are going to potential molecule we are going to identify. Then that molecule we are taking to the in vitro studies, enzymatic inhibition studies, in cell line studies. Then, based on that outcome, which one will be the best one? It may be a molecule, or if you know already existing molecule, it's fine. Suppose if the formula or any single plant, if there is no molecule till date it is reported, then we have to go to the bioassay guiding, fractionation, and isolation. You may be Dr. Anupam may be taken about the PRS, phytochemical reference standard, how we are going to the isolate, how we are going to develop the PRS, phytopharmaceutical reference. That they were going to isolate and they were going to give to us, or we will do the thing also, that same kind of the works. And we will find out how the molecule is working in the in vitro cell line studies and enzymatic inhibition studies. This is the one way. Then we will take which one is potential in that out of 10 screening, which one is giving a promising result, we will take into the in vivo studies. And now the in vivo studies we are doing, there is a rat, we can use mice, rat, and different kind of things. One more thing is now zebrafish. Zebrafish, now we are going to establish in our lab because zebrafish genome sequence, 70 to 80% genome sequence, it is equal to the human genome, and there is a lot of research is going in that, and it is easy to maintain, and it is very less time consumption to do the study, and all this reason now we are going to start our zebrafish lab also, the in vivo study. Once this is happen, then in tissues we will administer the active fractions on molecules, and we will study in the things, and based on the efficacy, then tissue we will take, we will study the tissue level, how it is, what are the molecular pathway it is going, how it is acting, mechanism, what is the possible mechanism like allopathic drugs we are going to establish. This is our primary setup. We We are taking this as a lead, but we are not going to, as such, we are going to follow. We are going to implement different kind of the strategies, and we are going to make it our Ayurveda Siddha Unitas, how it is producing a efficacy, and we are going to establish that molecular mechanism point of view also. In the gene level or any particular protein level, in cellular level, what are the pathways it is following. So, for example, in insulin pathway, how the glucose it binds, what are the inside of the cyclic pathway it is happening, how the blood glucose level is changing. So that kind of the establishing the pathway we are going to do the things. Next slide, please. So, like this, we are going to work on the network pharmacology also, but network pharmacology, uh, which will help us to provide single formulation how they were using for the different disease conditions. So where the target is coming, a different gene, different protein will act. For example, one formula is there. If you are looking at traditionally, they will be mentioned that it is used in the treatment of inflammation, diabetes, uh, hepatopathy, splenopathy, everything they will be mentioned. It may be a single herb or it may be a combination of the formulation. So how it is acting, because it depends upon the disease conditions. In the each disease it is having a different gene something formation. So it is going on diabetes, it is acting on a particular gene and molecular level where it is producing certain changes and restoring it. Similarly, in hepatopathy, it is going in such a manner. So, which is the pathway, which is the target that we are going to study in this. Okay, next slide, please. Yes.
There are herbal drugs that claims to have a gene regulation property,
Which regulation?
gene regulation. So
Uh,
it can regulate the transcription, translation of the
uh,
drug.
so, I don't think for
Gene.
gene regulation till date, no traditional medicine or anything, no one where it is used. So that aspect is entirely different. It may act on the gene, but gene regulation, I think no medicine till date it's not available. Only disease, but we can say that particular genetic sequence may be the effect, it may be how it is producing. That the genomic sequence also we can study. Disease control, things we can able to study. But drug, as you asked specifically for the gene regulations, may not be there. But in disease condition, how the gene has been things and in the treatment after the treatment, how is the genetic that sequence it is changing and what is the pattern we can able to study in that by using Western blot and other kind of genomic sequence analysis, where we can able to study in advanced level. We can able to study. So now, if you are looking, this is one example. Similar manner, we are going to work anti-inflammation. Means there is a number of if you are looking, there is around 10 plants where it is listed. What are the effective molecules? They may be, means effective molecule means these molecules isolated or they may be identified through the LCMS, GCMS technology. They may be analyzed and what are the genes. So the same molecule, it is acting in a, it's a, see, this molecule is nothing but whether it is in our phytochemistry or it is in our allopathic phase, all are chemicals. If you are isolating, if you are bringing it to the table as a powder, all are chemicals. The chemicals, if you are taking in a particular concentration, it will act as a drug. If you are overdose, if you are taking, ultimately it will act as a toxic substance. So these chemicals, it is acting in each individual gene or targets, receptor, which will make a different configuration because the molecule it is having some chemical structure, which is having a functionality group. The functionality group will have a amine, amine group, NH2, OH group, and chlorine, Cl, chlorine, where lipophilic characteristic will be able to get increased. But if the molecule goes to the particular target, these targets are proteins. Proteins are made up of amino acids. We know amino acids ultimately is a chemical moiety, chemical structure will be there, where individual the interaction will be hydrogen bonding, covalent bonding will happen due to the interaction of the molecule, the free atoms present in that the amino acids protein with that other chemical. So each molecule it is going to the each target. The each protein will having a different amino acid sequence, which may having a different interaction and which may lead to the intracellular level where there is a changes in the genetic pathway things. The pathway of the things it will be entirely will get different. So different therapeutic effect we can able to use. Curcumin. can able to use for the anti-cancer also and inflammation also, diabetes also, how it is acting. The same molecule. It's ultimately in the protein, what is the amino acid sequence and where it is binding pocket. In the same amino acid on the protein, there are different binding sites will be there. Which site it is going to be bind and what is the disease condition. Based on that, the efficacy will be produced. So that we are going to study in our next lecture. So these are some examples. So another one helps where they identified a different kind of the regulatory of the bone marrow and everything. So they identified different pathways and things. We are going to work this we can call as a network pharmacology. We will call as a network pharmacology, and we will get some data based on that. We can able to develop establish our efficacy point of view. Next, next thing, previous slide. Okay, what is the outcome if you are doing this system pharmacology approach? Scientific validation of the herbal medicine you are going to, or why you are using for the particular therapeutic use, you can able to study. And phytoconstituent responsible for the herbal medicine therapeutic use and role also will be established. Previously, we don't know which because of which molecule it is present in that it is producing. Simply we will say multiple phytoconstituents is present in that it is producing a synergistic activity, antagonism activity, and now we are going to prove that synergistic activity is there. Two or three more phytoconstituents acting in how the efficacy is there. So for that also we have to run a synergistic activity study also we will run. That everything is lengthy protocols and at the time of posting we will do something like that. Then molecular mechanism, which gene, which protein it is acting, multiple therapeutic benefit of the things we can able to establish. A repositioning of your medicine, herbal medicine to the new therapeutic use also there is a possibility to establish. If you are looking at network pharmacology, sometimes it will act on the medicine formulation where uh, the constituents previously it is used only inflammation and diabetes or hepatopathy. And network pharmacology time, if you are doing it, may act on the some cancerous gene also. So we can consider that we can try based on the things, we can try the anti-cancer studies, and if it is producing efficacy, we can establish the things for the new therapeutic use also the particular thing. Then composition of the multi-ingredients, multi-plant constituents, means plant used in that, we will come to know the things, and it will win the physician consumer confidence. So the consumptions of the herbal medicines will be rationalized scientifically. Yes, this medicine, if you are taking, it is producing effect due to presence of these plants. These plants, it is having a, these are the phytomolecules. These phytomolecules, it is acting in a this pathway, and hence where we are getting a efficacy.
Sir, have you identified any herbal plants that can cause cancer?
The cons can not a cons,
The,
but
that can
anti-cancer.
cause that can treat cancer. I mean, that can treat cancer. Have you identified or isolated any identified a specific herbal herbal plant that can treat cancer
Okay,
with
that
those?
will be useful in the cancer treatment. Um, See, first we have to go through even ancient literature. Ancient literature, if anyone has written that it is used in the treatment of somewhere, definitely it will be in our Ayurveda, that is authoritative books it is written. Or otherwise, the new positioning, the repositioning of the herbal medicine for new therapeutic use I have mentioned. No? You go back slide, go back. Now, if you are looking, uh, you can consider like this previous one, no? Previous one. Back, that diagram, back. Okay. Similar manner, the previous one slide was there, no? Okay. Now, if you are looking anti-cancer, okay. So this anti-cancer already they were using it fine. Now we consider that you don't the use of the anti-cancer, but the if you are going to network pharmacology, there is a certain kind of genes where we will select the things. We are going to make that a computerized. So based on the database, suppose it is showing it is acting this molecule, this plant constituents also acting on the particular cancer target gene. It is showing. Then we can look that and we can try whether it is having a anti-cancer activity or not. We will go to the in vitro studies, first in silico studies, because some target we know. So anti-cancer protein we will take, and computer where the molecule we will get a dog, we will see whether it is there. That validation we will go to the in vitro study. In vitro study mainly because of the cell line study, we will go and see. And if in vitro cell line studies, if it is yes, it is showing some promising, then we will take to the in vivo study. Then we can take to the top. So this network pharmacology will help to identify existing herbal medicines. It is apart from mentioning in our authority book any other new disease conditions whether we can able to use. The predictions we can able to, but the prediction only. You have to confirm with studies in silico, in vitro, in vivo studies to get it. This is the one way. Another way, tribal peoples where they were using the for cancer or such kind of the thing without knowing things. So if you are coming to know the things and you have to do the validation, you have to take the things and make a extract, go to the in vitro study, then in vitro studies, cell line studies, it is efficacious. You go to the isolation of the molecules, then you can go to the in vivo, then you can establish. It's a regular pathway you can establish. Okay, your query is clear, clear. So next one, next, okay. Now if you are coming to the top previous one. See now if you are coming to why the study is required, why the pharmacological study is required in the herbal. If you are doing the quality control alone, it is a fine. If you are doing the chemistry, microbiology, we are developing quality control, it's fine. But it is not efficacious, or it is producing any drug interactions. Even you can do the drug interaction study also. For example, the herbal medicine, if it is anyone, it is saying that the main backbone is what is the thing, no? Herbal medicines after the patients or physicians after the taking, or physicians sometimes they will say allopathic physician, but other physicians they will not say traditional physician. Okay, so if lack of efficacy anywhere it is reported, maybe safety issues may be reported. Herbal medicine, if it is showing some kind of a safety issue, it may be immediately brought to the notice. But any time, whether the lack of efficacy is reported anywhere, whether anybody is saying no, this medicine was either practicing physician or in case of the patients, they were nowhere it is reporting. The pharmacovigilance, it is not only reporting only the adverse drug reaction, it is also to report the lack of efficacy also. But till date, if you are taking the data, most of the time the lack of efficacy may not be reported at all, because physicians what they will do immediately they will change into the other molecule, they will go, they will not report in that. And traditional medicine people they will not say that yes the medicine is not working, and patient also it is not working, so let this physician is not good, we will go to the other physician. Okay, so lack of efficacy is not at all reported in anywhere compared to that of the safety. It may be reported, it may be percentage, maybe 5% only. It may be. Very rarely it will be reported. So now our aim is what the WHO is saying that this is the WHO for the quality control perspective. WHO has set a one thumb rule. The rule is one is the constant know about the therapeutic activity identified clinically if it is studied. Example, curcumin, reserpine, such a kind of the molecules, methionolide, such a kind of the molecules where you can herbal phytoconstituent you have to use as a quality control marker. You have to use as a quality control marker. There are different kind of the marker. Maybe Dr. Anupam may be taken chemical marker, negative marker, clinically, pharmacologically marker, then clinically things. So rule number two is saying that if there is no clinically effective marker, you don't know about a particular plant, due to the particular plant, this phytoconstant is responsible for the therapeutic efficacy. You don't know. At least you should know the pharmacological marker, recognized pharmacological activity. It is studied in vivo studies. Such a kind of the marker you have to use. If nothing is there, then you have to go to the chemical marker and other type of the marker. But nowadays, if you are looking, I think Chinese they were using that pharmacological marker and the clinical marker. But remaining all our system people, if you are looking other countries system, they were not using any of the clinical marker or pharmacological factor. Simply they were using certain kind of the chemical marker. For example, gallic acid, routine, quercetin, epicatechin, catechin, galactoepicatechin. All are reported in all uh, polyphenolic compounds in cannins. All are it is reported. But but specifically that is having on activity. But a particular plant specifically like a curcuma langa, where if you are looking at where the curcumin is an active constituent, similarly each is having the that particular clinical marker till date not yet identified. It is a very low number of the plant only identified. So our pharmacological, our role is to identify such a kind of the compound, to study the things. Even if it is the compound is not available, at least to rationalize that particular extract, which pathway it is working, where it is working. To rationalize things, we are going to work. Next slide, please. So the which means that next slide, please. Array, next slide, please. So our main role is to identify the constituents, active constituent recognized for the pharmacological activity that we are going to provide to the quality control sections. You can use this as a marker. Now, this part, now the PRS lab, phytochemical reference lab, they were going to identify they were going to isolate the compound and they were going to give to us, and we are going to study the pharmacological activity, and we are saying we are going to say that yes, this molecule is much better, working better. So they may be used that is for the quality control aspect in the future in the pharmacopoeia. This is our aim, like in case, but how it will take a long process, it will take time. Next slide. Okay, now the summary of outcome. What is the evidence based? So evidence based, this is a scientific evidence. We are working in the reverse pharmacology or system pharmacology model. We are going to work in that, identify the molecular mechanism, pharmacologically active compound we are going to develop for the therapeutic or clinical marker for the future drug development process, and strengthening the quality standards. So nowadays they were using only the chemical markers instead of chemical markers. Each formulation, these are the active ingredients. So these active phytoconstituents, the level how it is present depends upon the therapeutic efficacy also will vary. So that we are going to make future perspective. It is strengthening the scientific evidence on the molecular mechanism generating a safety and efficacy also. Now we know because of which constant efficacy is coming, which constant it is not efficacious. Simply present in that. Now we are going to make such a kind of the things whenever allopathic drugs taking concomitant medicine if they were taking as your questions, we can able to understand where the drug interaction is taken place. So that study also we have to do the things previously with in vitro, then later stage where we will go to the animal studies, then clinical. But that is the next step. Because the first step still date there is no clear cut molecular mechanism or is not yet established for many of the plant drugs. So that's where we are working in that aspect. Okay, so we can able to position our medicine in international market in a much better way. So apart from quality control alone, if you are doing your product can be used, but if with the combined manner efficacy study if you are showing, and this is the reason for the efficacy if you are providing, that molecule will be the best selling molecule. You will get an economic also, it will get boom, and your medicine also worldwide will be researched to that all the people. Otherwise, it will be within our country. Within our country, we will use, and our potential it may be there, but that other country we cannot able to take the things. Other humanity means other global populations, they are not going to get any benefit. Next slide, please. So now this is our actually we are doing in our lab at present stage. As I said that now we have there is a advanced computational molecular modeling software we are having. We are having a docking studies as well as a molecular dynamic studies, in silico studies, and phytoconstants we are doing. Where if you are looking for molecule, if you are looking the protein structure. So this is for example this for alpha glucosidase. Alpha glucosidase, you know, there is the enzyme is responsible for the breakdown of the disaccharides into the monosaccharide. Hence, there is a blood glucose level will get increased in diabetic patients. There is the agarose. Agarose, one allopathic drugs where it is available. The drug is responsible for inhibition of this alpha glucosidase enzyme. Because of it is the inhibition where the disaccharide will not be converted to the monosaccharide, and hence the blood glucose level is controlled. Okay. Now there are many herbal plants or drugs. It is they were saying that anti-diabetic activity. Now we have studied one molecule. That molecule is called as a cyanidine. It is well reported molecule in many plants. So cyanidine we have taken, and we where there is agarose bound with that alpha glucosidase. It is the enzyme. Enzyme is made up of protein. Correct, na? The enzyme also it is a protein. So in the enzyme it is available in that we can there is a public domain. There is a PDP protein data bank. it is available. In that where we have to take a disease with the target, so that enzyme that is where you are seeing that is the enzyme, the first one, alpha glucosidase enzyme, where in the lowest level, one small molecule like it is there, no, it's a bounded, it's bind with that. What we have taken already, agarose bound site, well known, they crystallized this protein, alpha glucosidase enzyme, so that we have taken, and where already agarose is bind, bound in that, we are removing that in that pocket, whether our phytoconstituents is acting with binding or not, and what are the binding it is taken place. Where the next picture, if you are looking the molecule, the green color, and if you are looking ASP, aspartate amino acid. Okay, so there is a number will be there. So threonine 204. So these are the amino acids. In this amino acids, what are the interaction? If you're looking, there is a dot line, it is there. So that's where hydrogen bonding, covalent bonding. So based on that bonding, how the interaction is taken place, the molecule is binding. Whether the similarity of the amino acids bound with the agarose, whether this is also same or any difference is there, and based on that there is a docking score will come. We will come to know which one is the best docking score. So whether agarose comparatively we can able to study. And next one, if you are looking at the next, that last one, just you can click, this is a molecular dynamic study, just click. नहीं, नीचे, हाँ, click that. No, yeah, go back. uh, that figure you can touch. How there is interaction is taking place inside of the molecular dynamic, how it is happening. You can come and see in my there is no issue. Okay, the next one.
Sure. In your modeling software, can we det- can we determine if a molecule which in where allosteric sites or uh, in the active site of an enzyme or
Yes, yes,
where it can dock?
yes. There is a
Uh,
different approach we can do. The first approach is already where there is some molecules it is bind, allopathic drugs if it is bind to that. In that same pocket we can use. Otherwise, a different pocket we can identify. Further, there is a one more analysis, sitemap analysis, one more analysis is there that we can do, and we can find which pocket is there, best pocket. In that pocket, we can able to dock it and we can find. There is no necessary. These our phytoconstituents will only need to bind onto the allopathic binding site. It can bind to any site. We can analyze protein, different pockets, different sites, and best site also we can choose that. No issue. There are two different ways. One is there is a constrained manner focusing, already existing pocket. Another one is without that flexible. Any pocket where it is going to be bind more, that pocket also we can identify. There are two way method we can able to do the docking. But we are going with, since allopathic drug standard is there, we are going to comparatively we are going to make that yes particular pocket we are whether it is acting or not. But we can do other pocket also.
can that also determine the type of inhibition or competitive, non-competitive
Uh,
inhibition?
no, non-competitive till date we have not studied that. That will be only we will do with the enzyme kinetic study in vitro study. In vitro study that we will do. That is our plan is there. Uh, Once the lab is established, we will do with the in vitro enzymatic kinetic study. We will do. That will be in vitro. And here till date I have not explored that area. Let I will ask with the software person whether it is there or not. Then I will. The next one is where the in vivo study, as I said that we are going to establish the zebrafish model lab, in vivo study, and molecular mechanism oriented study. So this way, I hope another 6 months our lab will come into the running condition. Zebrafish lab also. Next slide, please. So these are the things. In silico pharmacology studies, we are going to do the things. Network pharmacology, we are going to do the in vitro studies, as you asked, the enzyme inhibition study and its kinetic studies, we are going to do the things. And next one is in vivo pharmacology with the molecular pathway analysis, like a Western blot, the PCR, genomics thing. So such a kind of the studies also we will do. But now we are very initial stage. Hope another 1.5 year it will get everything. It will get properly set up. Will be next. Thank you. These are the references. If you want, you can go through it. Next. Thank you.
So may I ask what software are you using for the molecular model?
We are using well industry renowned software storing here.
That
Storing.
is the German.
German. The, use. Now the companies
So Schrodinger.
SCSRODING.
Okay, thank you.
About the Sidium guajaba here in India, as they claim that this is an agent for an anti-diabetic agent. The Sidium guajaba. Does it prove that this is an anti-diabetic agent? Do you have an uh, document about this? It was mentioned in the uh, during our tour in plant origin. Yeah, it's a plant origin. It's it's a raw plant. Psidium, Psidium.
PubMed Central, Science Direct. Definitely they may be published.
Sir, most of your herbal products fall under the category of food supplement. Mm, what's it? Okay.
Register to register and sale under food and health supplement point of view only they were considering.
Mm.
They were not accepting as a drug now.
And
Because
do you have combinations like uh, two or more combinations of the plant?
yeah, not is even on drug they said shaman crops they will say that it's around high it is there. There is a more than 10 plant combination also in our Ayurveda drug it is there. For asthma and other things where they were for general immunity purpose where they were using Chavanpras. So one a very well famous to Ayurvedic drugs where it is having around I think 40-45 plant composition it is there.
But if you have that combination of those herbal plants, how can we tell if that the um if it has a side effect, like how will how are we going to know if the side effect is from one particular plant if it is a combination?
Yeah, correct. Actually, no, traditionally, if you are looking, they were saying that they made the composition of different composition they were using because they were each one it is having a nullifying action also. One is acting suppose synergism point of view, another is acting in antagonism point of view. But holistic manner, we are in a holistic manner. We want to study in such a kind of things. Till date, it is nowhere it is reported or it may be known, but no one is reported. No one is reported the things also. So till then, it is considered as a safe. And if you want to do the study, then we have to do the detailed system pharmacology analysis, and even we can able to conduct some kind of the clinical studies, controlled clinical studies. Now they were patients are taking things and they were moving, and so it should be avoided. We have to do the like allopathic controlled clinical studies we need to do, and we need to find out. And if any study is already published, whether they reported any safety issues, also we can able to retrieve the data. But whether the studies are there or not, we don't know. Maybe in COVID time they will perform some kind of Chavan-class immunity perspective. They were saying that it is a immunomodulator kind of the formulation. So if they conducted any studies, you can search in the clinical studies. Uh, clinical studies, clinical studies of India. Some website is there. In COVID time, whether they done any studies, you can able to look that. In that, definitely will be there. But these now whatever product we are using, everything based on our traditional times, continuously the very long period they were using. I think there is no controlled clinical data also not available. That's where since the do the clinical controlled clinical study, it is a lot of time consumption and money also will be there. So that's why now know what is the possibility to explore the uh, efficacy or safety aspect, where some preclinical studies, in silico studies, where we can able to some toxicity studies we can able to perform and we can prove. But if you really if you want to know how the clinically it is producing a toxicity, then you have to any side effects, any adverse effects it is present in that. Definitely, you have to do the controlled clinical studies because regular time you cannot able to control the patients like he's saying. Nowadays, allopathic medicine also they will take, traditional medicine also they will take. How you will justify? How we can able to justify? Yes, the side effect is based on this or that, or how there is a interaction with this kind of case. We cannot able to say. And sometimes the continuously some of the patients will take, some of the patients will not take the traditional medicines properly. Maybe alternative days they may take, or 1 month if you are prescribing within a 15 days due to many factors, he may avoid the intake of the medicine. So you will not get enough result, safety, or what you are asking, side effects and all this. Controlled clinical trial based on our clear cut aim for what purpose you want to do that clinical study. Proper protocol we have to make and we have to do the study. Then we will get a data for the things. Until otherwise, traditionally it will be keep on it will be using that.
morning. So how you are feeling in India? Yes. Hope your gut microbiome is healthy. Yes. 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 Especially na. So when we change the continent or when we change the region or place, so basically our habits of the food also change. So will also affect our gut microbes. So whatever the microbes are we have in gut, our gut, so they affect our gut microbes also. Okay. Okay. Shall we continue? So just see this slide. We may see routinely newspapers, news channels. So recalling the products. So recalling the products due to some contamination issues, due to the exceed of the limits, or due to the aflatoxins in their presence, or due to some chemical constituents like ethyl glycol we have recently faced an issue. Okay. So we may face similar issue in this microbiology also. So these microbes are in everything. Okay. Before human beings, these microbes are available. Before human beings, the microbes are available. They are billions years of ago. So you can see this contamination of the products. So basically, the main principle of taking the drugs is for healing purpose. If the drugs only containing the microbes or any pathogenic organisms, there is no purpose in the using this drug. So before consuming drugs, we have to ensure so this drug is free from the pathogens and is healthy to drink or healthy to take. Next slide. So this scenario we face how we are in pre-COVID and post-COVID. So in pre-COVID, so we are free. So even uh, laymen also don't know what is microbes. But after post-COVID, even a vegetable seller, even a labor, they don't know. They didn't study also. They know little bit care about this microbes. They immediately they wash their hands with alcohol, sanitize their hands. So they get so much awareness, so much knowledge, or so much fear due to this microbes. So we know how this COVID is distract entire world. Okay? A single tiny virus we can't see with our naked eyes also, but how it affects our lives. Okay? So many deaths, so many financial losses, so many restrictions on moving okay, from one place to another place, contamination. So this is due to this microbes. Next. So dealing. So how we deal the microbes. Okay. From morning onwards and up to bed night. So we our minute every minute is deal with microbes. We just see without brushing we can't start our day. If you are not brushing your teeth, so your teeth will decay due to these microbes. Okay. Then after sleeping also before going to bed, you have to check your bed sheets are clean or not. So every minute, every second, we think about microbes. So in front of this water bottles are there. Okay. If I taken this water and give it to you, here to you, will you drink? Will you take this water? No, no, no. Because so I'm a healthy. Of course, I'm a healthy. But you suspect, okay, some may contaminated. So you avoid. Even our own family members also sometimes we avoid. So in every point, in every occasion, in every situation, we will deal the microbes. Next, will you say this the difference between the first figure and second picture? Will you say anyone? You can fill the feedback from left also. Okay? You can fill the feedback from left. Will you tell any difference between the first picture and second pic? Is there any difference? I think in your country also they have milk products. Yes, yes, Tetra Pak. Tetra Pak. So the first one is the packaging of the milk in quality, and second one is Tetra Pak. The first one is a we call pasteurized milk. We call it pasteurized milk. Pasteurized. Pasteurized. Wherein pasteurization, what they will do, they will boil the milk at 70 degrees centigrade for 5 seconds to 10 seconds. Okay, 70 degrees centigrade, 5 to 10 seconds. In Tetra Pak, they boil the milk at 140 degrees centigrade and higher temperatures. Okay, so in higher temperatures, there is no microbes. Okay, in low temperature, 70 degrees centigrade, they have the chances to regenerate the microbes, bacteria. Because bacteria will generate spores, so the spores may generate. Okay. That's why the shelf life for packed polythene package is 3 days. They give shelf life 3 days. But this Tetra Pak they give 6 months. It's a liquid. It's a liquid. Both are liquid. Both are liquid. But this Tetra Pak they give up to 6 months. You can keep it room temperature also without opening. So that's the difference. Just 6 months Tetra Paks you can keep in normal room temperature. Tetra milk, Tetra Pak milk you can keep in normal room temperature up to 6 months. Once you open, you have to consume immediately. But without opening that Tetra Pak, you can keep 6 months room temperature. No refrigerator, nothing. We can extend. No, no. It's, they give maximum 6 months. You can use also up to 6 months, more than 6 months also. But the deterioration will be started. So that's why they will not recommend. They give up to 6 months shelf life. They give. So the main difference is that is pasteurization. This is sterilization. So sterilization means completely removing the microbes. Completely removing the microbes. Whatever we are using ophthalmic solution, it should be a sterile. It should be a sterile ophthalmic solution. So that's the main importance of the sterilization. What is the importance of the microbes in this traditional medicines? Okay. So we know this traditional medicine, 70 to 80% from the plant source. 70 to 80% from the plant source. These plants are originating from the naturally. So they contaminate with microbes. Okay. Due to soil, due to water, due to air. So we can expect microbial contamination. 100% microbial contamination with plant material. These are naturally originating. Okay. And this is important on the cultivation also. How they are cultivating, where they are cultivating the herbal medicines. So we'll take next slide. See this. This is leave this one. These four factors are very important in the quality of the herbal medicine. Good agriculture practices, good harvesting practices, good manufacturing practices, and good laboratory practices. See, good agriculture practices. So basically, in anywhere, you will take so much care on commercial products. Well, rice, wheat, okay. Any commercial product we will give daily, we will give it irrigation water, we give pesticides, we will apply herbicides, okay. Everything will take care. But coming to this cultivation of hair, no one can take care. Okay? They let them go, then they will go and they will collect and they will transport. Okay? No one will take care. Okay? They will not give proper irrigation also. If any insects, any pests attack, no treatment. So good agriculture practices are also important. And some uh, herbal medicines is also grown in industrial area where this industrial effluents are pumped out, fluid. Okay. So there may be chance of contamination with heavy metals. Okay. That effluents are is full of heavy metals, so that we may expect okay, that herbal medicines also contaminated with heavy metals. Next, good harvesting practices. Okay. So where they harvest the herbal materials and how they are transported to industry. So from north to south means we have 2,000 kilometers. The industries are located somewhere and the cultivation is somewhere. How they are transported? So they're transporting in a normal temperature at 37. So microbes will also generate at normal temperature. If they're transporting a raw material in a freeze container, okay, the temperatures are controlling, humidity factors are controlling. So we can control the microbial population. So this good horticulture practice is also very important. Next, good manufacturing practices. Good manufacturing. So after reaching the industry, okay, how they're starting processing the material, raw material. If they are washing with deionized water or not. Okay. If they are treating properly or not. Okay. Good hygiene. Everything is comes under good manufacturing practices. Okay. And their facilities, their lab facilities, in-house facilities for testing all the parameters, safety parameters, including microbial, chemicals, everything. Next, good laboratory practices. So after finishing the product, you have to check the quality of this product. If your lab is not a good enough facilities, good equipment, so how you will test the parameters? Okay. And good laboratory practices is, for example, in a microbiology lab, this room is fully contaminated with microbes. I'm, I'm testing this. You just imagine it is a sample we received for testing. We testing the microbial quality of the sample. We've seen so many microbes. Oh, we have so many microbes, but our lab is also already contaminated with microbes. So the, we will not justify our practice or whatever we are testing. So in this scenario, our lab should be a aseptic condition. We should maintain aseptic conditions. Then we can perform this test. Okay, then we if we see any microbes on this sample, then we reject this sample. Yes. So this is a good laboratory practice. Next, so these are the basic instruments required for the microbes, microbial laboratory. Okay. So autoclave. Uh, so I said autoclave is for sterilization purpose. So first one picture I shown sterilization. So in this autoclave, it is a normal pressure cooker. So it works under steam under pressure. So steam under pressure, including spores, will also die. Bacterial spores will also die. Then hot air oven, biosafety cabinet. So now I will show this all in our lab also. So anybody dealing now microbes, microbial from you? Okay, another lab. You are from food only, na? From food FDA. So in food also you also test food. No? So microbiology also they have food microbes. Also. Yeah. Next, the sample preparation. Okay. So generally, whatever this microbial analysis, not only similar for the not only for the herbal materials, it also similar for the food also. Food and allopathic drugs also. Okay. Same methods. Sample preparation. So there are different samples are there. If you just imagine it is a sample we received for testing, it is a herbal medicine. Okay, we received for testing. If it is just freely water soluble, So we can just pick your 10 ml and then we can go for testing. If it is a water solid. Next. See, some products are fatty nature. They have so much fatty nature, so it is very difficult to dissolve in water. So in this case, we will add queen 80, the surfactant. So when we add this queen 80, so it is very easy. So fatty product for this purpose also we add polysorbate 20 for this product. So then it will be easy. So this method is called sample preparation for the microbial. Method suitability test. Okay. So just imagine as a regulatory point of view, you went to some visited some industry, okay, and you collected the samples for testing, quality testing. You have to test whether these samples are good or not as per the regulation. Okay. Then you have to test this sample in microbiology point of view. First, you assume this medicine or this drug is free from the antibiotics or antimicrobial agents. If it is contain antimicrobial agents, so obviously we not found any microbes. So that's the first we have to check whether this uh, sample or drug is free from the antimicrobial. For this purpose, we just add whatever the pathogens we are testing into this sample. If this medicine or if these drugs promote the growth of the microbe, okay, we can assume this drug is not contain any antimicrobial agents. If it is not showing the any growth of the microbe, so this product contains antimicrobial agents. Okay. So then we have the other treatment. So we have to pre-treat the sample. Then I can go for that. So these are the inhibitory agents. Uh, if we found any inhibitory agents in this our drug samples, we have to add this type of uh, neutralizing agents. Next, growth promotion test. So our purpose is any microbes are present in this drug sample. We have to test whether this growth is promoting or not. So we have to conduct a growth promotion test. And also we'll keep one negative control also, only buffer solution for this negative control. Next. So these are the medias, different medias we'll use for the uh, isolation and identification of the microbes. Media. Okay. So our nutrient purpose, we will take so many nutrients. We will take carbohydrates, phosphorus, potassium. We will take so many nutrients. Similarly, for the microbial growth also, we have to add, we have to give this type of nutrients in the form of media. These are called culture media. Next, these are the specific medias we use for the specific pathogens. For microbial limit test, we use buffer sodium chloride peptone and casein soybean digest agar, saburu dextrose agar. For Escherichia coli, we use lactose broth, nutrient broth, selenite broth, McConkey broth, McConkey agar, and leuciniacin methylene blue agar. For Salmonella, we use tetrathianate billion green broth, bismuth agar, deoxycholate agar, XLD agar, and urea. For Pseudomonas aeruginosa, we use citramate agar, uh, Pseudomonas agar, and mannitol. So, you know, E. coli, Salmonella, Pseudomonas aeruginosa, Staphylococcus aureus, these are highly pathogenic organisms. So this should be absent in our drugs. Whatever the herbal medicine we are consuming, these pathogens, these four pathogens should be absent. So why only these four pathogens? There are so many pathogens, thousands numbers of pathogens are there. But only four pathogens. We have Shigella, we have Listeria, Vibrio. There are different pathogens are there, natural pathogens. How are we come tell why this only four pathogens? See, microbial limited. Okay. So as I told, uh, these herbal medicines are from the natural sources, plant material. Okay. So we will not expect sterile products. Okay. We will not expect this plant material is completely free from the bacteria. No, we will not expect that because they are naturally sources, so they are contaminated with bacteria. That's why we'll give some limits. Okay, certain limits we can accept, we can consume. It is good for our health. No, it is no harmful. It's called microbial limit. Microbial limit is a quantitative elimination of bacteria and fungi that grow under aerobic conditions, and it's a qualitative test for the absence and presence of the specified microbes. It's called microbial limit. So there are different methods are there. Membrane filtration method, total plate count method, and most probable number method. So based on our sample, we can choose any of these three. Wait, T1 is total yeast and mold. Yeast and mold. I will tell you. See, this is a membrane filtration method. Okay. So in this method, we can take the 45 microns filters. Okay. Then we will pour this. This is a drug, our drug, herbal drug. So we'll transfer this drug through this filter. Okay. Then this filters trap the if any bacteria contain this sample. Then this filter will keep on a medium. Then we'll keep in incubator 37 degrees centigrade for 2 to 3 days. For fungus or yeast and mold, we keep for 5 to 7 days. After incubation, if we see any colonies, growth of the bacteria, then we can count how many colonies are present on, how many bacteria present on this sample. This is called membrane filtration. Next, this is called total plate count method. Okay. In total plate count method, there are two methods are there. Okay. Pour plate method, spread plate. Pouring. Okay. So this is our drug sample. We received a drug sample for testing. Okay. Take 1 ml. Okay. And you just imagine this is a agar plate, nutrient plate. Okay. Then we can pour this 1 ml. Then we pour media on this. Then you can stir it and keep for incubation. It's called pour plate. In spread plate method, we add this media to this petri plate. Okay. Then take 0.1 ml on this. Then we can spread with the spreader. This is called spread plate and incubation. For bacteria, generally we use soybean casing. For yeast and mold, we use potato dextrose agar. And for bacteria, we keep uh, 24 to 48 hours incubation. For fungal and yeast, we keep 5 days, maximum 5 days. Because fungus will grow very slow. So we keep up to 5 days. Next, this is most probable number method. It is a statistical method. It is not an accurate method. So we mostly will not use this method. We mostly use filtration method or total plate count method. So Eastern mode. Yes, ma'am.
There in our manufacturing plant,
Yes.
we have before we schedule, if we have a scheduled production,
Yes.
we have to
Check the
prepare the
quality.
place. The microassay department will conduct a swabbing or an open plate method, yes, to ensure
Microbial quality.
microbial if it's okay na. If it's not, if it will not uh, if there's still if there is too numerous, if the bacteria is too numerous to count,
Yes.
then we will again sanitize and disinfect the
Yes.
everything in the room
Yes.
from the ceiling to the floor. We do that, sir,
So that
there
is
in our
in manufacturing. So like I told previously, good manufacturing practices. Okay. So good manufacturing practices, we have to enter our field where we are production is there. It should be free from the microbial machine also, and there is a limit also. Particular limit we can accept. For example, it is 4 5 bacteria using. Okay, we can carry forward. 4 5 is very less. If you see more than 5, so we have to stop that and we have to take the precautions. So we have to fumigate or we have to sterilize, we have to do aseptic any swabbing. Okay. Or you have to use methyl alcohol to clean a wipe every surface areas, contamination. So these precautions, these measures we have to take. It's called troubleshooting or aseptic conditions. So for yeast and mold. So for yeast and molds, similar method. So have to take this is a drug sample. Okay. You take the 0.1 ml and SDA medium, Sobro dextrose agar medium, and pour 1 ml and you spread it and keep in incubator for 4 to 5 days. After 4 to 5 days, if you see any growth, any colonies, then you can count and you can distribute. Next, see, this is our example. Okay, this is a 10 to the power of 1 dilution. This many of bacteria you see on plate in 10 to the power of 1 dilution. What is 10 to the power of 1 dilution? See, this is a sample. Okay, so this sample may contain a huge number of pathogens, huge number of bacteria. Okay, so I'm taking directly 1 ml from this sample and pouring on this plate. It is very difficult. You see how much population is there. So I can't count how many. I cannot tell how many population is there. For this purpose, we will dilute this. It's called serial dilution. So we take serial dilution method. We have to take 4 to 5 test tubes with 9 ml of sterile water. We take 1 ml from the sample and transfer into first test tube. It's called 10 to the power of 1. Means we are diluting 10 times. 1 ml diluting 10 times. 10 to the power of 1. You see 10 to the power of 1, we got this many products. Okay. From 10 to the power of 1, again we take 1 ml to next test tube. It's called 10 to the power of 2. 100 times we are diluting. So similarly we are diluting, so we are reducing the microbial population. So it is very easy to identify the bacterial colonies how much we have. Next slide. You see, is the reducing. Next. See, next this is the 10 to the power of 3 slide. Next. So 10 to the power of 4, see how we can easily count bacterial colonies. This many of bacteria is present. Next. See 10 to the power of 5, you can count easily. See 10 to the power of 6, so 20 to 30 colonies are there. So it is very easy to count how many colonies are in this sample. This sample contains this many. This is the one formula, colony forming unit, bacterial colony forming unit. So this is a principle we can apply if it is in 60 colonies in 10 to the power of 2 dilution. So our total bacteria in the sample is 16 to 10 to the power of 4, means 60,000. 60,000 colonies are present in this sample. 60,000 bacteria are present in this sample. Now we have to check. Now we have to see our regulations. our pharmacopoeias, how much bacteria standard, how much bacterial limits are they are accepted. So different pharmacopoeias have different limits. For India, next this is for India we have, for example, for total aerobic plant we are accepting 10 to the power of 5, means 1 lakh. Now our sample is got 60,000. So it's within the limits. So we can accept this sample. We can pause this. If it is a more than 1 lakh 20 per of 5, we can reject this sample and we submit the report to the concerned agency. They will recall the entire batch. So, for example, in case of East End mold, 10 to the 3, 1000 East End mold only permitted, only 1000. This four specific pathogens: Staphylococcus, E. coli, Salmonella, Pseudomonas. These four should be absent. These four should be absent. So these are for the herbal extracts and powders. So next tea. Now previous slide I told we have taken only four specific pathogens: E. coli, Salmonella, Pseudomonas aeruginosa, and Staphylococcus. Why means these are antibiotic resistant bacteria. These are mostly antibiotic resistant bacteria. You know the word escape. Just remember this word escape. These are escape pathogens. Okay, escape: Enterococcus, Klebsiella, Enterobacter, Acinetobacter, Staphylococcus, Pseudomonas. These pathogens are most antibacterial resistant bacteria. See, this antibacterial resistance is a silent pandemic. It's a silent pandemic. Okay? So it is more more lethal against the coronavirus. We don't know how many deaths are going on this antimicrobial resistance. We have here one government approved MTCC, Microbial Type Culture Collection Center. MTCC, MTCC, Microbial Type Culture Collection Center. So they give certificate. They give certificate, and they main they may huh, if they want, if your country need that cultures, you have to root through this biological diversity. NBA, we have one national biological diversity. First, you have to approach them. Then they give clearance. Then you will get that cultures. It is a different procedure. This culture, per type cultures. These are similarly ATCC cultures. You know, American Type Culture Collection Center. They will same maintain this here in India. MTCC cultures, Microbial Type Culture Collection Center. Here in Chandigarh, it is in just 3 hours journey from here. One street there. That is also government organization, central government organization. They are maintaining this MTCC. See, this antimicrobial resistance is a serious pandemic. So we don't know how they are dying, how they are this spreading through man to man, okay, generation to generation. Okay. So we need just in 2000 up to 2010 only the last antibiotics are discovered. You just see the history of the antibiotics. In last 2000, 2010 only the last antibiotics. Okay. And whatever the pathogens and whatever the antibiotics chemically synthesized, these organisms are showing resistance. Okay. Whatever the drugs we prescribe, for example, we prescribe in Staphylococcus aureus, in the first instance we prescribe penicillin. But penicillin also, this organism showing resistant to penicillin. Then they change to methicillin. Then methicillin also showing the resistant this one. So when we be prescribing the drugs, antibiotics, it's very important whether these pathogens are antibacterial resistant or not. See, one example, if you are admitted in hospital, first we have to know the cause of the infection, whether which pathogen is there and it's showing the resistance or not. Then only proper administration of the drugs will be suggested. Next. See, these are the WHO guidelines for microbial limits. You see, aerobic microbial count also they given 10 to the power of 5 and yeast and mold they given 10 to the power of 3. Similarly, in our Ayurveda Pharmacopoeia of India, in India also they are maintaining almost we are maintaining same standards as similar with the WHO. But in WHO, they given one extra two pathogens, Shigella and Clostridia also. But we are not testing these pathogens. And this is European Pharmacopoeia. You see the similarly we are maintaining 10 to the power of 5 and 4 for these standards. This is Indian Pharmacopoeia. So in Indian Pharmacopoeia also we have similarly 10 to the power of 5 and 4. Okay. And they are also given Shigella and Salmonella. Next. USP, United States Pharmacopoeia. So there they are treated as a dietary nutrition supplements, botanicals and dietary nutrition supplements. So they are almost similarly same standards. It is Brazilian Pharmacopoeia. In Brazilian Pharmacopoeia, they treat it as a vegetables. In herbal drug, they treat as a vegetables. So they are given the standards almost similar to the one API. In Japanese Pharmacopoeia also similarly, almost similarly, we are maintaining standards in Japanese Pharmacopoeia. Next. In Australian also they have Therapeutic Goods Act. So in that also they given the microbial acceptance criteria for traditional. So they also given similar 10 to the power of 5 and 10 to the power of 5 for total aerobic microbial count and 3 for yeast and so it's a comparison uh, between different pharmacopoeias. So we have compared the Indian pharmacopoeia, European pharmacopoeia, British, okay, Japanese, Thailand. Okay, these are have same 10 to the power of 7 and 10 to the power of 5 and United States pharmacopoeia and WHO Brazilian pharmacopoeia they have the same. Next. One article we published recently on this microbial elements and herbal drugs and preparation. We are comparing almost 14 pharmacopoeias, international pharmacopoeias, and we are made this article. Coming to Escherichia coli. Okay. So just we discussed that this these pathogens are more antibiotic resistant bacteria. So we have to identify if we sample received this sample. So we have to identify whether this sample contain the Escherichia coli. Okay. So Escherichia coli is a gram-negative bacteria. Okay. You know gram-negative, gram-positive. You know gram-negative bacteria, gram-positive bacteria. See, based on our morphology, so we categorize them men and women. Okay, men and women. Okay. So based on the morphology of bacteria, they divide it to gram-positive bacteria, gram-negative. Okay. And this step is very important. We have for identification of gram-positive or gram-negative. Based on that only, we have to prescribe the antibiotics. We have to use the antibiotics. See, if you use penicillin antibiotic on gram-positive bacteria, it will not work. So that cell wall morphology is different. So it won't work. So that's why you have to check whether it is a gram-positive bacteria, gram-negative. See, for identification of Escherichia coli, uh, see first test tube you see it is a called McConkey broth. It is a violet color. Okay. If your sample have Escherichia coli, it turns into yellow color. Yellow color. Okay. And you see the bubble formation also. Down bubble formation. So we can confirm 30%. Okay, my sample, this sample contains teacher issue. 30% we can confirm. Then after that, next slide. Then after that, you add a indole. Indole is a reagent. Okay. When you add a indole, it form a red color ring. So 50% we can confirm our sample contains equal. Next. Then what you will do? Then from this sample, you seek on a McConkey agar. It's a McConkey agar. So you just see the red colonies, pink to red colonies. Okay. Okay. 70% we can confirm this sample, this drug sample may contain. Next, from this plate, you can streak on a EMBR. Using methyl and blue. You just see the uh, colonies shining. Okay, transmitter when you show on the light, so it's like blue black appearance. So we can confirm 100%. We can confirm this sample is contained. So accordingly, we can give the next. You are working online. Hello, ma'am. You work from home. You have work from office. Okay, go back. Next, salmonella. So salmonella, generally you listen, he is suffering with typhoid. So due to this salmonella, typhoid. So this is also mostly contaminated with water. This is also gram-negative bacteria, waterborne pathogens. Even Escherichia coli also waterborne pathogen. So salmonella also gram-negative bacteria. Okay, it is also very serious pathogen and it is also antimicrobial resistant pathogen. So if we sample, we have to test. So we transfer into that tetrothianate billion group. Okay. Next, let's see. If your sample contain salmonella on bismuth sulfate agar, you see the black column. Okay. So this black only means this bismuth sulfate contain hydrogen. Okay. Will supply in that media potassium tellurate. When this potassium tellurate oxidize and they produce the hydrogen cyanide. So the black colonies are, we can see, okay, we can confirm it is a solvent. Next. So 70% we confirm that is a salmonella. Next confirmation, we use a triple sugar iron. Okay. So that red one is a control one. When we inoculate that plate colony into this test tube, you see the gas formation and it turns into entire black. Okay. Hydrogen cyanide production. Due to the hydrogen cyanide production, it turns into the black. So we can confirm, okay, this sample contains Pseudomonas aeruginosa. Pseudomonas aeruginosa is opportunistic pathogen. Mostly we can hospitals, okay, when we go to the hospitals, it will predominantly spread. It is also gram-negative bacteria. Okay. So for this, we have a specific media. If you spread this sample on this citramide agar, it will produce pigments, green color pigment. Okay, you see the first plate pigment, and you can keep under UV light. You see the fluorescence. How they give the fluorescence under UV light. Okay, we can assume it is a 70%. It is a pseudomonas aeruginosa in our sample. Next, for confirmation, we can do this oxidase test. Okay, these are oxidase discs are there. When we keep the our sample on this, okay, it will turns into the violet color. It is a positive. Okay, the sample confirm this is Pseudomonas aeruginosa. Next, next catalase. So catalase is a when aerobic organisms. So you, Just see, we are all aerobes. In bacteria also there are aerobes are there, anaerobics are there. It will not tolerate, anaerobics are not tolerated to oxygen. If you supply the oxygen, it will death. But this Pseudomonas aeruginosa is an aerobic organism. You just add a few drops of hydrogen peroxide, H2O. Okay, it will give the bubbles. Bubbles. So the catalase enzyme release, okay, H2O to the Blake and superoxide dismutase, and it form the bubbles. Okay, we can confirm this catalase test is positive for Pseudomonas aeruginosa. 100% we can confirm this sample contains the Pseudomonas.
Are available here.
Which one? Ma'am?
All the all the agents that you are
Ah,
using,
we have. We have.
we have.
Yes. We will show.
Because there in the Philippines, we sometimes find it hard for some agents. We sometimes find it hard to look for suppliers, and it's really very costly,
See,
very expensive.
H2O2, H2O2 is available in any medical store. We use for our when we got infection, the doctors also use H2O2, nitrogen peroxide.
Oh no, it's available anywhere in the Philippines. Some of
Nice.
their agents, some of
Reagents
their
also
agents
available.
that we
Yes,
use are
yes, we have so many industries, manufacturing industries.
agar-
In media also they give ready-made media. Okay. Yeah. We have so many facilities in see. We don't need to carry individual tests. Okay. They some manufacturer they supply a kit. Okay. You just pour the sample on all the wells. You can keep 24 hours. You will get the results. You just calculate and you submit the report. The kits also there.
for our microassay procedure, we wait for 5 days.
Yes. For bacteria, 24 to 48 hours. 2 days for bacteria. Bacteria will go speed. For yeast and mold, we have to wait for 5 days. Fungi, yeast and mold, we have to wait for 5 days. That is incubation. Bacteria, 2 days will go. Bacteria will go. Yes.
It passed or it failed.
Wait.
So that's the
So up to 5 days, you will wait for the report.
both
Mandatory.
the both the powder,
Mm.
the raw material,
Raw
and
material.
the finished product.
Yes. Next, Staphylococcus aureus. So Staphylococcus aureus is a gram-positive bacteria. Okay. It is mostly skin infections. Skin infections. You just see skin infections. You see the pus forming bacteria. It is a pus forming. You see the pus on your skin. So it is due to Staphylococcus aureus. Pus is due to the Staphylococcus aureus. So if you want to check the Staphylococcus aureus in our blood samples, just use streak on X-ray. See, it's a mannitol salt agar medium. First one is a control. It's a red color plate and it completely turns into the yellow color. So we can suspect it is a Staphylococcus aureus. I'm not understanding.
The right side is the passive
Yes,
active.
this right side is the positive one, and the left side is a control. It's a control. Okay, without sample, it is a control. The red one, and the yellow one is a positive for Staphylococcus aureus. So for confirmation, we will take this bacteria and we streak on another medium. Next, see, this is called Bayer Park agar medium. See, black colonies with white zone. The white zone is there, and center black colonies are there. Black, black. Okay, we can confirm it is a Staphylococcus. Next. So up to this, these all are the for the Ayurveda, Siddha, Unani drugs, and this for the homeopathic medicines. I think in your country they are using homeopathy also. Homeopathy also. There are the regulations are in okay. Okay. So basically, this homeopathy drugs are prepared from the ethyl alcohol. They are mostly they use alcohol for preparation. They make the dilution. They make to up to 1,000 dilution. They describe. Okay, this generally we think this homeopathic medicines are prepared in mother for ethanol or ethyl alcohol. So this microbial grow. But most of the homeopathic medicine they use 40% ethanol. When we use 70% ethanol, then only they act as an antimicrobial. When we use 40%, it will not show any antimicrobial. They use cosmetic. They, we can use 40%, okay, no issue. But it's more than 70, using an
What's the difference between homeopathy and allopathy?
yes. Homeopathy is a different concept. Okay. Allopathy, you know what you are using chemical formula. Okay. See, antibiotic, you say penicillin. You know beta lactam. Beta lactam ring. You can know this drugs, this beta lactam is present in your drug. Coming to homeopathy, so in homeopathy, they prepare from the plant origin, chemical origin, insect origin. They prepare from the plants, animal, snake. They prepare from the snake, reptiles, okay, spiders, lizards, cockroach. Okay, they prepare from more chemical origin also. They use heavy metals also, like arsenic album, drug we use in COVID time. From heavy metals also we prepare the homeopathy. In homeopathy drugs, what they will do, they will prepare a mother tinctures. Okay. For example, they take the plant material, okay, 10 grams plant material, they keep in 100 ml of ethyl alcohol, 100 ml of ethyl alcohol. Okay. And this mother tincture, they dilute into 10,000 dilution, and they do the potentizer. They have a potentizer machine. They will beat like this medicine, and they assume that it will increase the power of the medicine, and they will dilute it. Okay? Then they will 6cx potency, 12c potency, different dilutions. Allopathy is clear. You can know functions, you know clinical trials, everything is there in allopathy. But homeopathy, finding the which content is there in your medicine is different. How you will found in 10,000 dilution your product, very different. Which chemical constituents are there, which phytochemical constituents are affecting. It is somewhat different. In homeopathy, one other interesting products also there, nosodes. Over here, nosodes. Nosode means it's similar to vaccines. Nosodes. Okay. Nosodes also in India they use not only in India, in so many countries they are using nosodes. Okay. Similarly, they treat as a vaccines. Vaccines. Yes. Okay. So what they how they prepare nosode? See, yes, nosode. Yes, sterility test for nosode. Nosodes. Okay. Nosodes. So how they prepare? See, nosodes also prepare from the bacterial pathogens. Okay. For example, this is a Mycobacterium tuberculosis. You know TB. So TB is caused by mycobacteria. Okay, mycobacteria. Okay. So they grow the mycobacterium. Okay, then they kill that mycobacterium, sterilize and they kill the mycobacterium, and they sterilize and they filter it. Okay, then after filtration they will do the dilutions. Then they use as a method. Similarly, in vaccine also they will do same thing. But the vaccine we know the action of the antigen, that anti-toxide what we are using in vaccine. We know how they are affecting, how they are killing the, how they are boosting the immunity, how they're triggering the antibodies in our body. But in also that science is lacking. It's a difference between the source and next. So in homeopathy also we do sterility test for ophthalmic solutions, and this source will do sterility test whether the presence or absence of any material. We have to see that the turbidity. So these three media is used for the source sterility test. The value we have to take for the testing. This much value we have to take for the testing. These are of standards in our homeopathy pharmacopoeia of India. So we have four systems. Four systems have different four pharmacopoeias. Ayurveda pharmacopoeia, Siddha pharmacopoeia, Unani pharmacopoeia, homeopathy pharmacopoeia. We have four pharmacopoeias. Next, preservation of microbial cultures. See, these madam, you asked, these are the from the MTCC culture. Ma'am. So these are the MTCC cultures. So we will we will take from them and we will maintain up to 5 years. Then again we will again freshly we will take from them. Within 5 years we will take from them, and we maintain this specific cultures for our reference culture. We will reference culture. We will play करो. इसको PPT से बाहर आकर play करो. See, it is a fumigator. Okay, fumigator. Okay, right now, madam has told we have when we are facing contamination, when you found population in your place before proceeding to the manufacturing. So, it's a fumigation is a good method. So we have to fumigate the entire room before proceeding. So you can fumigate weekly. No, machine very less. See, for of course, if you don't have machine, if you permitted, you can use formaldehyde and potassium permanganate. Okay? It is a very good formaldehyde and potassium permanganate. Okay? If you नहीं तो बाहर से प्ले करो, प्ले करो. Otherwise, I will show in our lab. See, if you sure दो. So this, what we'll do, we will every Friday we will do this fumigation our lab. Okay? Then after coming Monday, we'll keep empty plates and we'll see the quality of our room. Uh, Exposed to just open the plate, open the plate, and we'll keep for 6 hours. Then we'll see how what is the how this affecting our room. When more populations are there, we will take more serious concern. Okay? So, so in every microbiology lab, in every industry, fumigation is very important. Then we can control the microbial. It's a very easy method. Next. Thank you. Thank you very much for your attention. So now we will go to our lab. Do 2, 3 hands-on experience. Later also you can you have more time. after lunch also you will use.